Hi, my name is Melissa Kelly, and I am a genetic counselor at the Geisinger Genomic Medicine Institute and a member of the ClinGen Education Workgroup. Through this presentation, I will provide a brief introduction to the use of population data in the ACMG and AMP sequence variant interpretation guidelines. Population data is when a large cohort of people, or a combination of multiple smaller cohorts, is used to understand a variant's frequency in the general population. Historically, this type of data was assessed using small cohorts of unaffected controls, but we now have much larger data sets to evaluate. The variant's frequency is referred to as its allele frequency, which simply means the number of times that variant or allele was identified, divided by the total number of alleles or chromosomes tested. For example, if the variant has been seen as heterozygous in 20 individuals, or 20 alleles, out of the 10,000 individuals tested, or 20,000 chromosomes, the allele frequency is 0.1%. If the variant is uncommon, this frequency can be referred to as the minor allele frequency, or MAF. So how is population data helpful? Well, it allows us to identify which variants are common and therefore generally thought to be benign for traditional Mendelian disease from those that are rare or even absent from these large data sets. It is important to remember that rarity does not automatically mean that a variant is pathogenic or disease-causing, only that it is uncommon enough that it is possible. Population data also allows us to compare the frequency of a variant in affected individuals to the frequency in unaffected individuals or in the general population. When a variant is seen more often in affected individuals, it starts to suggest that the variant is contributing to disease. If the difference in allele frequency between cases and controls is statistically significant, we can say that the variant is enriched in that group. Currently, the availability of large population data sets allows us to understand the frequency of a variant with more granularity and in many instances has invalidated prior control studies that only tested 100 or 200 individuals. For example, a published study might have concluded that a variant was pathogenic because it was not seen in 100 controls, but our current larger data set shows us that the variant is actually present at a low frequency. But while population data has provided many benefits for accurately assessing variant frequencies, there are still important limitations to keep in mind while interpreting this type of data. They include population sample size, the ethnic groups that are included, the phenotypes that are included, and the testing technologies used to generate the data. A more detailed description of these considerations are provided separately in the Introduction to Population Data presentation available on the ClinGen YouTube channel. In 2015, the American College of Medical Genetics and Genomics, or ACMG, and the Association for Molecular Pathology, AMP, published a joint consensus recommendation for standards and guidelines for the interpretation of sequence variation. The overarching goal of these guidelines, in addition to providing a consistent standard, was to create a framework to determine if a variant within a gene that has a definitive role in a Mendelian disorder is pathogenic for that specific disorder, as well as to reduce the number of variants that are reported as being disease-causing that have insufficient evidence. It is important to note that this framework, as it is written, is not designed to consider variants in genes with minimal evidence supporting their association to a Mendelian disease, which are often referred to as genes of uncertain significance. To accomplish this goal, the guidelines define 28 criteria that address various types and levels of evidence that either support a pathogenic classification for a given variant, with levels ranging from supporting to very strong, or support a benign classification with supporting strong or standalone evidence levels. This figure provides a visual of the different criteria outlined as part of this guideline and how they are grouped into different types of evidence. For this presentation, we will be focusing on population data, which is the top row of the figure. As previously mentioned, population data can be used with different levels or strengths of evidence, which are outlined at the top of the figure, and the table to the right shows how different combinations of evidence can be combined to determine the final classification. For example, a variant can be classified as pathogenic based on one very strong and two moderate criteria being met, or by having two strong criteria being met. A variant could be classified as benign if the standalone frequency criteria is met, or if two strong benign criteria are met. As we go through the different uses of population data in this framework, it's important to keep in mind that each criterion we discuss will have a different level of evidence, and to keep in mind how that relates to the specifics of that particular criterion. Population data is useful in assessing a variant's potential pathogenicity by looking at the frequency of the variant in a control or general population. Depending on how common or rare the variant is, that can be used as evidence to either support that a variant is too common to cause disease or is rare enough to potentially be pathogenic. There are 5 criteria in the ACMG AMP guidelines that look at variant frequency, which we will go through in more detail. To determine a variant's frequency, one can search publicly available population databases, such as the Broad Institute's Exome Aggregation Consortium, or EXACT, or the Genome Aggregation Database, GNOMAD, as well as using race-ethnicity matched control data that is published in the literature. It is important to consider what ethnic groups or phenotypes each source includes when evaluating a variant's presence or absence. Variants can be common in one ethnic group and absent in others, so to ensure that the variant is not common in the ethnic group of interest, that group needs to be sufficiently represented in the population database. Otherwise, it remains possible that the variant is common, but was just not seen in the population database due to that ethnic group being absent or underrepresented. To introduce how an allele's frequency supports that a variant does not cause disease, we will start with the strong benign criteria. In general, when a variant's frequency in the general or control population is greater than what is expected for the disorder, it is considered strong support for a benign classification for a rare Mendelian disorder, or criterion BS1. Essentially, what this means is that the variant is too common to be a cause of disease. One way to think about this is by comparing a variant's frequency to the most common pathogenic variant. If we consider variants in cystic fibrosis, also known as CF, as an example, the incidence of disease is 0.031% in Caucasian individuals, while 3.6% of individuals will be carriers. Roughly 1/3 of all carriers, or 1% of all Caucasian individuals, will have the most common pathogenic variant, a deletion of phenylalanine at codon 508, also known as delta F508. Therefore, if you are considering a new variant in CFTR, the gene for cystic fibrosis, that has an allele frequency close to or higher than 1%, it is too common to be a cause of disease. An example of this is a variant at codon 75, a change of arginine to glutamine, which has been seen in 2.5% of Europeans. This provides strong evidence that this variant is benign, because if it was pathogenic, it would be the most common pathogenic variant for CF. Additionally, if a disease under investigation is fully penetrant at an early age and the variant is observed in a well-documented healthy adult individual as homozygous for a recessive condition, heterozygous for a dominant condition, or as hemizygous for an X-linked condition, then this is also considered strong evidence for a benign interpretation, or criterion BS2. An example of this would be finding a novel homozygous variant in PAH, the gene for phenylalanine hydroxylase deficiency, or PKU, in an adult with a normal diet who does not exhibit any of the clinical features and has normal enzyme activity. If the variant was pathogenic, some phenotype is expected. For X-linked disorders, it is important to consider if the variant has been seen in hemizygous males or in heterozygous females. It is also important to keep in mind that use of this criterion is dependent on the disease of interest and its penetrance or variable expressivity, as well as knowing how well the population or control cohort has been phenotyped. Now that we've introduced how an allele's frequency supports that a variant does not cause disease, it is hopefully easier to understand how a higher allele frequency provides stronger evidence that a variant is benign. In general, an allele frequency over 5% is considered a standalone support for benign classification, depending on the size of the population tested. This is a very conservative frequency cutoff, and by taking into account the prevalence and penetrance of a specific disease, as well as the genetic and allelic heterogeneity, lower thresholds can be considered. Exceptions to this do exist for some common pathogenic variants, and laboratories will adjust allele frequency cutoffs accordingly to be able to detect these variants. Currently, the ClinGen Sequence Variant Interpretation Working Group, SVI, and multiple other clinical domain working groups are creating specific recommendations and guidelines on how to adjust this standalone and other frequency-driven thresholds, using disease and gene-specific evidence. Additional recommendations from these groups can be found on their respective web pages on the ClinGen website. Switching now to evidence that supports a pathogenic role. If a variant is absent from a large general population, or in the cases of recessive diseases and genes, is below the expected carrier frequency, and the population is race ethnicity matched to the patient with the variant, then this observation can be considered a moderate piece of evidence for pathogenicity, criterion PM2. For some diseases and genes with reduced penetrance and variable expressivity, where it is expected that pathogenic variants would be present in these larger population databases, a threshold for absence or rarity can be determined based on prevalence, penetrance, and heterogeneity estimates for that disease gene combination. Because many benign variants are also rare or considered private, so unique to an individual or a family, absent in a race ethnicity matched population is considered a moderate piece of evidence for pathogenicity, but it is not currently considered sufficient to be used as strong evidence. When assessing for a variant's frequency in population databases, using large multi-ethnic populations will be the most informative compared to using a smaller ethnicity-matched control group, since the variant may actually be more common in a different ethnic group. Lastly, as with evaluations of any population data, it is important to consider the limitations of the database being used. For example, if the variant is absent from next-generation sequencing data, one should confirm that the read depth is sufficient for an accurate call at the variant site versus appearing absent due to a lack of coverage. It is also important to keep in mind that population data for insertions or deletions may be poorly called, which is an established limitation of next-generation sequencing. To be able to use population data as strong evidence of pathogenicity, additional stringency above being absent from population studies is required and is predominantly assessed using case control comparisons. When the prevalence of the variant in affected individuals is significantly increased compared with the prevalence in controls, criterion PS4 can be used. The use of population data for these comparisons is most informative when both populations are well phenotyped, have large frequency differences, and the Mendelian disease under study is early onset. The significance of these comparisons can be calculated using odds ratios or relative risks. Odds ratios are a measure of association between a genotype, essentially a variant, and a phenotype, essentially a specific disorder or a particular outcome, and can be used for either Mendelian diseases or complex traits. An odds ratio of 1 means that the variant does not affect the odds of having disease, while values above 1 mean that there is an association between the variant and the disorder, and those Odds ratios below 1 mean that there is a negative association between the variant and the disorder. The confidence interval around the odds ratio is also as important as the odds ratio itself. If the confidence interval includes 1, then there is less confidence in that association. When trying to calculate an odds ratio, it's important to remember that in general, patients referred to clinical labs for testing are likely to include individuals sent to rule out a disorder and thus may not qualify as really being well-phenotyped cases. Also, as previously mentioned, when using a general population as a control cohort, the presence of individuals with subclinical disease is always a possibility for diseases with late onset, reduced penetrance, or variable expressivity. And lastly, well-phenotyped case control studies are generally rare for most Mendelian disorders. Therefore, the observation of a variant in multiple unrelated affected individuals, combined with its absence from controls or population databases, is worth noting, and it may be used as evidence for pathogenicity at different levels of strength depending on the disease and gene being evaluated. So to review again, population data is genomic variant data from a large cohort of people used to understand a variant's frequency in the general population. It can help differentiate common variants from those that are absent or rare, as well as identify variants enriched in affected individuals. But it is important to remember that all of these resources also have limitations. The ACMG AMP guidelines can use population data in combination with other types of evidence to arrive at a classification for a given sequence variant. There are 3 criteria that provide evidence towards a variant being benign, which are generally focused around variants being too common to cause disease, and there are 2 criteria that provide evidence towards a variant being pathogenic, which are generally focused on variants being rare enough to potentially cause disease, and that can be combined with case data to support a variant's role in disease. Lastly, I would like to again highlight that the ClinGen Sequence Variant Interpretation Working Group, or SVI, and multiple other clinical domain working groups are creating specific recommendations and guidelines on how to adjust these frequency-driven thresholds using disease gene-specific evidence. Additional recommendations from these groups can be found on their respective web pages on the ClinGen website. I hope this has been a useful introduction to the use of population data in the ACMG and AMP sequence variant interpretation guidelines. Please visit www.clinicalgenome.org for additional educational resources and to learn more about ClinGen initiatives. Our funding sources are listed at the bottom of this slide. Please feel free to contact ClinGen by email at clingen@clinicalgenome.org with questions or feedback. Thank you for your time.